Hello friend, welcome you all in this session. I am BP Shah, PGT Bio, Kendriya Vidyalaya Kesapuram. Today I am going to continue the topic respiration in plant in session 5, chapter 14 of class 11th of NCERT. In my last session, I have asked some question. Now, I think you all will be able to answer these. First of all, look at the question which I have asked in my session 4 lecture, that is think over questions. The first question was, what is the main steps in aerobic respiration and where does it takes place? My second question was, which enzyme is required for the synthesis of ATP? Next question was, which type of respiration result in the formation of water? Now, these are the answer of those questions. What are the main steps of aerobic respiration? Where does it takes place? As we have already known, there are two type of respiration, aerobic and anaerobic. Aerobic respiration, it takes place in presence of oxygen, and there are various type of steps and their place where it takes place. These are the main steps are, first of all, glycolysis, where the glucose molecule change into pyruvic acid. The site of its occurrence is cytoplasm. Next step is Krebs cycle, which is also called TCA cycle or citric acid cycle. It takes place in the matrix of mitochondria. Third step is electron transport system. It is also takes place in inner mitochondrial membrane, and last step is oxidative phosphorylation, where ATP is produced. It is found in oxysome, where this oxysome is divided into two parts, FO and F1 particles in the inner mitochondrial membrane. So, this is the answer of question first. Now, question number second is, which enzyme is required for the synthesis of ATP? As the term enzymes, AAC is added at the end. So, the enzyme, the answer is ATP synthetase, which help in the formation of ATP with the help of ADP and PI, that is inorganic phosphate. My third question was, which type of respiration result in the formation of water? Two type of respiration, that is aerobic and anaerobic. In anaerobic respiration or the fermentation, there is no release of water. So, the answer is aerobic respiration. Now, look at this session start from the after Krebs cycle, that is terminal oxidation, which include two main steps, that is electron transport chain and oxidative phosphorylation. So, these two combinedly called terminal oxidation. So, we can define as terminal oxidation, it is the final step of the aerobic respiration which involve in release of proton and electron from the reduced coenzyme, that is the reduced coenzyme NADH2 and FADH2, and their ultimate reaction with oxygen, and finally, this oxygen react and for metabolic oxidation. It is also called better to know terminal oxidation because it occurs at the end of metabolic reaction of aerobic respiration. So, it is better to say it is called terminal oxidation. Now, it involves direct reaction between the oxygen, proton and electron to produce metabolic water. The NADPH2 in the form of this NADH plus H plus, it dissociate to form NAD plus proton and electron. FADH2 it dissociate to form FAD proton and electron. This proton and electron finally combined with the oxygen, it form water. Now, terminal oxidation, as we have already said, it has two steps, that is electron transport chain and oxidative phosphorylation. As the term indicates, it is a chain of electron that help in the transport from one place to the another place. Now, first of all, electron transport chain, it is a group of coenzyme and cytochrome that takes part in the transport of electron downhill along the increasing redox potential with loss of energy at each step. So, the electron transport chain is a chain of different type of coenzymes and cytochromes. At each step of electron transport, we will see electron donor, one molecule donate electron has less affinity for the electron than the next electron acceptor. At each step of the electron carrier, it include flavin, iron sulfur complex, quinone and cytochrome. Most of them of the electron carriers are the prosthetic group of protein, that is quinone and other cytochromes, in which the quinone are the highly mobile electron carriers. The inner mitochondrial membrane possesses 5 complexes. These electron, it contains 5 complexes are complex 1, complex 2, complex 3, complex 4 and complex 5. These complexes are complex 5, it is directly connected with the ATP synthesis. The ATP synthesis takes place in oxysome. This oxysome we have already dealt in last classes, it has 2 particles, that is F0 and F1 particles. These are the, you see here, these are the different type of complexes in the electron transport chain. Here the complex 1, this one, here is the complex 2, here is the complex 3, this one is the complex 4. Finally, the complex 5 are present here at the end of the 4. This complex 5 finally connected with the ATP synthesis with the help of F0 and F1 particles. The complex 1, it is also called NADH Q reductase or NADH dehydrogenase complex. It has 2 prosthetic group, that is FMN flavin mononucleotide and FES complex. Both electron and proton passes from the NADH to FMN. Here this complex is which is called NADH Q reductase or NADH dehydrogenase complex. It transfers the electron, it is made by FMN and FES complex and transfer the electron from one complex to the second complex. Now, the second complex of electron transport chain, it is called succinate Q reductase complex. The second one, this one is called succinate Q reductase complex, and the third one complex is called QH2 cytochrome C reductase complex. It has 3 component, cytochrome B, FES complex and cytochrome C. This cytochrome C complex made by cytochrome C1 and cytochrome C, it is combinedly called cytochrome C complex. So, this complex called QH2 cytochrome C reductase complex, and the fourth one complex, this one fourth one complex is called cytochrome C oxidase complex, which contain A3, A and A3, cytochrome A and cytochrome A3 complex, which possess copper ion at the center. The latter help in the transfer of electron to the oxygen. From here complex 4 we have already told, from the complex 4 the electron transfer to the complex 5, which help in the ATP synthesis, and this electron help in the formation of metabolic water with the help of oxygen. Now, respiratory chain, all these chains cytochrome complex are located in the mitochondrial membrane, and the individual of membrane members of the electron transport chain are electron carrier, because this carrier help in the transport of electron and proton from one complex to the another complex. Now, there are the different types of electron carrier and their sequences are, these are the 7 electron carriers which functions in specific sequence are. These electron carriers are NAD, FMN, coenzyme Q, you will see here these are the NAD H plus, this one FMN, this one ubiquinone or the coenzyme Q, here the next one cytochrome B, next cytochrome C, cytochrome A and cytochrome A3. So, these are 1, 2, 3, 4, 5, 6, 7. You can remember it, there are the 7 electron carrier which function in specific sequence, that is NAD, FMN, coenzyme Q, cytochrome B, cytochrome C, cytochrome A and cytochrome A3. These are the sequence are very specific 1, 2, 3, 4, 5 and 7 finally, and this sequence is going to be continue and electron transfer from one carrier to the another carrier. During the electron transfer, the electron donor get oxidized. The electron donor which donate the electron to the next one, this become oxidized, and those which receive the electron are going to be reduced. During the electron transfer, the electron donor get oxidized, while electron acceptors get reduced. So, this transfer involves redox reaction, because in these cases oxidation and reduction both are takes place. So, it is called redox reaction, and these are catalyzed by the enzyme that is called reductase. Now, how these electron are going to be transferred? So, we can summarize here. We have seen that there are the many steps where the electron and proton are released. Let us summarize in my last lecture. In glycolysis, 2 NADH and H plus is formed, where this in between the glyceraldehyde 3 phosphate, when it going to form the next 1,3 bis phosphoglyceric acid. In this step 2 proton is going to be released, 2 hydrogen is released, and this hydrogen is accepted by NADH plus H plus. In oxidative decarboxylation, where the pyruvic acid transferred to the Krebs cycle or the mitochondria, this process is called oxidative decarboxylation. In these cases, pyruvic acid form acyl coenzyme and form 2 NADH plus H plus. In TCA cycle or Krebs cycle, 6 NADH plus H plus is formed, and the site are isocytric acid, when it form oxalosuccinic acid, in these cases 2 hydrogen is released and this 2 hydrogen is accepted by NAD plus and form NADH plus H plus. And next one, alpha ketoglutaric acid when form succinyl coenzyme 2 hydrogen is also released, this one is accepted by NAD plus and form NADH plus H plus. And last one, malic acid when changes into oxaloacetic acid, this also form 2 hydrogen which is accepted by NAD plus and form NADH plus H plus. So, these are the 3 1, 2 and 3. At 3 step 2 hydrogen is released and this 2 hydrogen for NADH plus H plus. So, these are the 2 molecules of isocytric acid, 2 molecules of alpha ketoglutaric acid, 2 molecules of malic acid involved in this process. So, there are 6 NADH plus H plus is formed in the TCA cycle. And in one step I have already discussed, at the time of conversion of succinic acid into the fumaric acid, when succinic acid converted into fumaric acid, the 2 hydrogen also released, this 2 hydrogen is accepted by FAD, flavin adenine dinucleotide, and it converted into FADH2. So, in TCA cycle 6 NADH plus H plus is formed and 2 FADH2 is formed. This all NADH plus H plus and FADH2 now going to oxidative phosphorylation. The terminal oxidation, it is the final step of aerobic respiration, that is the end point of aerobic respiration, which involves to release of proton and electron from the reduced coenzyme, that is NADH plus H plus and FADH2 and their final reaction with oxygen which form metabolic water. It is known as terminal oxidation because it occurs at the end of the aerobic respiration. Now, it involves the NADH plus H plus FADH2 which are formed during the Krebs cycle, during the glycolysis, during the oxidative decarboxylation. It is going to be break into NAD plus, proton and electron, FADH2, FAD, 2H plus and electron. Finally, this proton and electron it combine with molecular oxygen at the terminal point of the electron transport chain, it form water. Terminal oxidation has 2 main steps, that is electron transport chain and oxidative phosphorylation. As the term indicate, electron transport chain, it is the transport of electron from one place to the another place, and oxidative phosphorylation, as the phosphorylation we have already defined in many times, there is the formation of ATP, here oxidation of molecule and the ATP is formed. So, in that reaction ATP is formed due to the oxidation. Now, electron transport chain, it is a group of coenzymes and cytochromes. It is a group which are made by different types of coenzyme and the cytochrome that take part in the transport of electron downhill along the increasing redox potential with loss of energy at each step. At each step of electron transport, electron donor has less affinity for the electron than the next electron acceptor. What is the group of different types of coenzyme and the cytochromes? These groups are NAD, FMN coenzyme Q cytochrome B, cytochrome C1, cytochrome C, cytochrome A and cytochrome A3. These are the group of coenzymes, these are the coenzymes nicotinamide adenine dinucleotide, flavin mononucleotide and the coenzymes, these are the cytochromes. So, electron transport chain, as we have already discussed, it is a grouping of coenzymes and different types of cytochromes, and coenzymes are there. These coenzymes we can easily NAD, FMN, COQ, cytochrome B, cytochrome C1, C, A and A3. Now, this in each step of the electron transport chain, the electron carrier include flavin here, iron sulfur complex, quinone and the cytochromes. More than one prosthetic group of proteins, it contains more than one prosthetic group. Quinone, this quinone, it is highly mobile electron carriers. These electron complexes, electron transport chain complexes have divided into 5 complex. These 5 complex are complex 1, complex 2, complex 3, complex 4 and complex 5. There are 5 complexes of chain are present in electron transport chain. This terminal complex, that is complex 5, it is connected with the ATP synthesis. It is found at oxysome, as we have already discussed. This oxysome has head, stalk, tail. This one head is designated as F1 and tail as FO. So, complex 5 involved in the ATP synthesis. Now, the other complexes are the first complex, that is the complex 1, it is called complex 1 NADH Q reductase. It is also called NADH dehydrogenase. So, the complex 1 is called NADH dehydrogenase. Complex 2, it is called succinate Q reductase complex. this one first one complex, this one second one complex. The third one complex is called QH2 cytochrome C reductase complex. It has 3 components, that is cytochrome B, FES complex and cytochrome C1 complex. The third complex, that is cytochrome C reductase complex, is made by cytochrome B1, FES complex and cytochrome C1 complex. And the fourth one complex, that is called cytochrome C oxidase complex. It contains cytochrome A and cytochrome A3. When we know all 5 type of complexes, we can easily solve the how the electron transfer from the one complex to the another complex. The structure of complex are of this type, that is in electron transport chain the complexes are formed in this form. Here the phospholipid membrane, it is the intermembrane space. This one is the mitochondrial matrix and this one position the inner mitochondrial membrane. So, entire electron transport chain in this figure divided into 3 main spaces: intermembrane spaces outside, middle spaces where the complexes are present just like the structure of plasma membrane, that is inner mitochondrial membrane, and inner one side, that is the mitochondrial complex. Now, as we have discussed, 4 type of complexes are there. Here the position of complex 1, which receive the proton and electron here, and this is the position of second complex, this is the position of third complex, this is the position of fourth complex. Here the position of complex 5, which ultimately react with the proton and molecular oxygen, which help in the formation of water. So, this is the complex, different types of complex. When we know the different types of complex, we can easily understand the phenomenon of electron transport chain. Now, the electron transport chain, which have 7 types of electron carrier. These electron carrier, as we have already shown in the last figure, that is NAD, FMN, coenzyme Q, that is also called ubiquinone, cytochrome B, cytochrome C, cytochrome A and cytochrome A3. So, we can easily write in this way in the simpler form. Here is NAD, that form NADH plus H plus, here is FMN, here is ubiquinone UQ, here is cytochrome B and here is cytochrome C, here is cytochrome A3, sorry cytochrome A and cytochrome A3. In simplified system of the terminal oxidation, now we can easily see that at 3 steps ATP is formed. In between FMN and ubiquinone, ADP when combined with inorganic phosphate form ATP. FMN receive the proton or electron from the NADPH, while the ubiquinone directly receive the electron and proton from FADH2. So, there are the two routes for electron transfer. One route goes from FMN and another route goes from ubiquinone. Finally, these electron transfer to the cytochrome B, then cytochrome C. In between cytochrome B and cytochrome C, ATP is released. From the cytochrome C, the electron transferred to the cytochrome A, and from cytochrome A, it transferred to the cytochrome A3. In between that, when electron transfer A to A3, energy is released. This energy is utilized for the formation of ATP by the combination of ADP and inorganic phosphate, it forms ATP. Finally, from the cytochrome A3, the electron combined with molecular oxygen, it forms water, that is called metabolic water. Now, this is the same thing. Here you will find that there are the two pathway of the electron transfer. One is here, we have already written here, that is the route 1. These are the route 1 stage. From the route 1, the reduced substrate, that is reduced substrate means NADH plus H plus and FADH2. These are the reduced substrate. This transfer their electron and proton to the NAD. This NAD now converted into NADH plus H plus, and then the electron and proton transfer from one cytochrome to the another cytochrome. This is the route 1. In the second step, this is the route 2. In route 2, where the FADH2 is formed, this FAD which I have already discussed here, the succinic acid in Krebs cycle, when succinic acid converted into fumaric acid, these two hydrogen molecule is released. These two hydrogen molecule are combined with FAD and form FADH2. This FADH2 transfer their electron or proton to route 2, that is directly through the coenzyme Q, not NAD. So, the FADH2 transfer their electron to coenzyme Q, then the same pathway of route first. So, there are the two route 1 transfer their electron or proton to NAD, that is at different step where the NADH2 H plus is formed, transfer their electron and proton to route 1, and in one step where the FADH2 is formed, transfer their electron to coenzyme Q, and later on both pathway is common, that transfer their electron to cytochrome B, cytochrome C1, cytochrome C, cytochrome A and cytochrome A3. So, these are the 7 carrier molecules. Finally, from the A3, that half molecule of oxygen combined with electron and proton form the ATP. So, we can summarize here the now ATP account is, which I have already discussed, when glucose transfer their electron to glucose 6 phosphate, there is the loss of 1 ATP. When fructose 6 phosphate to 1,6 diphosphate, loss of 1 ATP. 3 phosphoglyceraldehyde, 3 PGA, it form NADP, NADH2. This NADH2, here the 2 molecule of NADH2 is formed. So, 1 NADH2 which we have already discussed here, 1 NADH2 it gives 3 ATP, 1 here, 2 here, 3 here. So, this NADH2 gives 3 ATP, 2 molecules are there, so 6 ATP is formed here. At this step 1,3 bis phosphoglyceric acid, when converted into PGA, 2 ATP is formed directly, and here At 2 PEP phosphoenol pyruvate, pyruvic acid is formed, 2 ATP is formed, and in these cases pyruvic acid to coenzyme, here 2 molecule of NADH plus H plus is formed. So, 6 ATP is formed here. In Krebs cycle, in this step isocytric acid, oxalosuccinic acid, 6 ATP is formed. Alpha ketoglycic acid, succinyl coenzyme, 6 ATP is formed. Succinyl coenzyme to succinic acid, that is the substrate level oxidation, 2 ATP is formed. When succinic acid transferred to the fumaric acid, it is accepted by FAD and it converted into FADH2, 2 molecule of ATP is formed. So, total 2 into 2, 4 ATP is formed. Malic acid when converted into oxaloacetic acid, it is accepted by NAD plus and for 6 ATP. So, when we count the total ATP, that is 38 ATP is formed, but it was found that in most of the eukaryote cell 2 molecules of ATP are used for the transporting of NADH plus H plus produced in the glycolysis into the mitochondria for further oxidation. Hence, net gain is 36 ATP. So, when we summarize, total NADH plus H plus is 10. So, this 10 gives 10 into 3, 30 ATP. This 30 ATP, we find that the total NADH2 is 10 NADH plus H plus at the this NADH or H plus formed by glycolysis, by Krebs cycle, by oxidative decarboxylation. So, total NADH plus H plus is for 10. So, 10 into 3 we can finally summarize, that is called 30 ATP. 2 FADH2 is formed. So, this 2 FADH2 it gives 2 ATP, 2 into 2, total 4 ATP is formed. In substrate level phosphorylation, 2 ATP is formed in glycolysis, where the net gain is 2 ATP. So, 2 ATP at glycolysis, and 2 ATP are directly formed in Krebs cycle. So, total 2 ATP. So, this is the different steps where ATP is formed. This 38 ATP from NADH plus H plus, this 4 ATP from FADH2, this 2 ATP from glycolysis, this 2 ATP from the Krebs cycle. So, total is 38 ATP. So, when one molecule of glucose is going to be oxidized, it gives 38 ATP. This 2 ATP is used in transporting the acetyl coenzyme into the mitochondria. So, 32 minus 2, the total is 36 ATP is formed. So, it is also called the 38 ATP are formed in kidney, heart and liver, and 36 ATP are formed in muscle cell and the nerve cell. Now, here is the summarized form which I have already discussed. Glycolysis 2 ATP directly, in glycolysis 2 NADPH 2 6 ATP. When pyruvic acid changes into the acetyl coenzyme, 2 molecules of NADH2, 6 ATP is formed. In TCA cycle 6 molecule of NADH2, total 18 ATP is formed. 2 molecules of FADH2, 4 ATP is formed. 2 molecules of GTP when converted into ATP, 2 ATP. Okay. So, total 38 ATP, we can say that is the 36 ATP when the 2 ATP is consumed during the glycolysis and when transfer the acyl coenzyme into the other form. So, now we can summarize the electron transport in the form of these are the there are the 4 types of cytochromes are there, that is cytochrome 1, cytochrome 2, cytochrome 3, cytochrome 4 and cytochrome 5. These cytochrome help in the transport of electron from one cytochrome to the another cytochrome. Here you will find that this is the just like the plasma membrane like structure, that is the phospholipids are there, and in cytochrome this NADH it converted into NAD plus plus H plus and H plus is released here. This proton then transferred to the cytochrome 2 from the quinone, and from the cytochrome 2 it transfers to the cytochrome 3. Here H plus is formed, then cytochrome C, then it transferred to the cytochrome 4 and cytochrome 5 that help in the formation of ATP. This one here is a mitochondrial matrix are there, this one here the intermitochondrial membranes are here. So, we can summarize the ATP form. ATP is formed, first ATP is formed when the electron transfer from FMN to ubiquinone, 1 ATP formed here, from cytochrome B to cytochrome C, 1 ATP is formed here. Then electron when transferred to cytochrome A to cytochrome A3, 1 ATP is formed. Now, this proton from the mitochondrial matrix it reaches to the terminal end and it combined to the electron and proton from here it form water, that water is called metabolic water. So, now we are able to say the total number of ATP form 38. Now, these are the various type of stage where the ATP is formed and where these ATP are used for the further reaction. Now, the physiological value of the fuels are different types of fuels. We know that the glucose molecule when oxidized 1 gram glucose molecule it gives 4 kilocalorie energy. When 1 gram fat is going to be oxidized, this 1 gram fat it gives 9.0 kilojoule energy, about this is about 2.25 times more than glucose, but for the oxidation of fat it requires more amount of oxygen. So, it can't be oxidized easily because due to the presence of less oxygen. Fat gives more energy, no doubt. For the breakdown of fat it requires more oxygen. So, in the lower classes we will discuss that the camel in their hump it store fat, because the fat gives more energy in comparison to the glucose. In case of protein, 1 gram protein is going to be oxidized, it gives 4 kilocalorie energy. So, we can compare glucose and protein, it equally gives 4 kilocalorie energy, but fat gives more energy. So, it is better to store fat for the longer time. So, camel and the some other animals also store fat for the storage of food in lieu of glucose and protein. All the respiratory fuel directly glucose, fat and protein it combinedly enter into the Krebs cycle and the cycle is going on. So, we can discuss here the physiological value of fuel as well as the efficient how the our cellular respiration in the living system is efficient. So, the when we use in the lab condition the efficiency is more, but in the normal cases the efficiency is low, that is 45%. Now, we can totally summarize after this chapter, you can think over this. What are the net gain of ATP molecule per hexose during the aerobic respiration? We will discuss in case of anaerobic respiration in our previous classes. In anaerobic respiration, the total ATP is used, total ATP is for, that is only 2 ATP. Now, we are able to answer these questions. The total net gain of ATP at glycolysis, Krebs cycle, electron transport chain or oxidative phosphorylation, we can say combinedly, that is the terminal oxidation, the total ATP is for. You can find and summarize here, we will discuss it in later classes. Now, the next question, that is which process makes direct use of oxygen. We have already discussed that the oxygen is used, there are two type of respiration, no doubt, aerobic and anaerobic, on the basis of use of oxygen or in absence of oxygen. So, in presence of oxygen, that is called aerobic respiration, and this aerobic respiration gives more amount of energy, that is 38 or 36 ATP, or the another type of respiration, that is anaerobic respiration, that gives 2 ATP and are used no oxygen. So, it is called anaerobic respiration. Now, the third question is the last electron acceptor of electron transport chain during the oxidative phosphorylation is. Which one is the last electron transport chain during the oxidative phosphorylation? Here we can say that the electron transport chain are made up of the electron carrier as well as cytochrome. So, there are 7 types of electron carrier, and the electron carrier or the cytochrome, the last one, these 7 are we have already discussed here, that is we can find out your answer. So, these 7 types are different form, that is NAD, FMN, here is your answer, and finally, the last electron transport carriers are, that is when the electron transferred to cytochrome A2 A3. So, the last electron acceptor, which one you find, which one is the terminal step of the electron transport system. Now, before next question is, before combining with the substrate oxaloacetate या oxaloacetic acid, pyruvate changes into. This is the most fantastic question. Before combining with the substrate oxaloacetic acid, oxaloacetic acid is the first step of the Krebs cycle. It is combined with the acetyl coenzyme. This acetyl coenzyme formed by the pyruvic acid, which is the end product of the glycolysis. This pyruvic acid not directly enters into the mitochondria. So, first of all, this pyruvic acid converted into the other form, the reaction is called oxidative decarboxylation. What are the product of the oxidative decarboxylation, which result to enter into the Krebs cycle. So, name this product, that is the end product of the oxidative decarboxylation. Now, my next question is the number of glucose molecule required to produce 38 ATP molecule under the anaerobic condition. It is the question to think that the number of glucose molecule produce 38 ATP, that 38 ATP is formed in aerobic respiration, but here is the anaerobic conditions are here. So, you can find out in anaerobic respiration only there is the formation of 2 ATP. 2 ATP for how much glucose molecule is involved? The total number of glucose molecule involved to form in the anaerobic respiration is 1. So, 1 glucose molecule for 2 ATP. So, how much glucose molecule is involved to produce 38 ATP molecules under the anaerobic condition. So, this is the my question to think over the at the end of this chapter. So, we can summarize that total chapter here. The respiration, which is known as slow oxidation of the organic food that gives energy. Now, the respiration are of two type, that is aerobic respiration and anaerobic respiration. Two types of respirations are there in the living organism. Aerobic respiration, which takes place under with the help of oxygen, and anaerobic respiration, which is also called fermentation. There are two types of fermentation, it depends upon the where it takes place, that is in case of muscle cell, that is lactate fermentations, in case of yeast and other bacteria, that is alcoholic fermentation. So, two types of fermentation that takes place in, it depend upon the where it is going on, that is the lactate fermentation and alcoholic fermentation. Now, we can divide the after the fermentation in aerobic respiration, the whole thing which we can summarize in the step wise. In aerobic respiration, glycolysis is the common pathway, where all the substrate like glucose molecule it converted into pyruvic acid. This is also called EMP pathway, because this glycolysis, the entire steps of glycolysis was given by 3 main scientists. What are the name of scientists? That is Imbaden, Meyerhoff and Parnas. All first letter combined from Imbaden E, Meyerhoff M, Parnas P. So, glycolysis is called EMP pathway. The entire steps of glycolysis takes place in the cytoplasm. Now, the result of glycolysis, that is pyruvic acid. Now, this pyruvic acid, when oxygen is available, it undergoes aerobic respiration. When oxygen is not available, oxygen are not present in that particular system, it undergoes anaerobic respiration, that is called fermentation. In fermentation 2 ATP is formed, in aerobic respiration, that is 38 or 36 ATP is formed. So, when it undergoes aerobic respiration, the next step, the pyruvic acid converted into acetyl coenzyme. This acetyl coenzyme now enter into the mitochondria and it combined with the oxaloacetic acid. This oxaloacetic acid, it combined with the acetyl coenzyme, it forms citric acid. The first compound in the Krebs cycle, that is called citric acid, This citric acid, it has 3 acid group, that is COOH group. So, this is called TCA cycle, tricarboxylic acid cycle. This was discovered by Krebs. So, this cycle is called Krebs cycle, or the first product is formed, that is called citric acid. So, this cycle also called citric acid cycle. So, we can say the glycolysis, after the glycolysis the oxidative decarboxylation takes place. After the oxidative decarboxylation, the acyl coenzyme enters into the mitochondria. This mitochondria further goes in mitochondria, Krebs cycle is going on. In Krebs cycle 6 NADH plus H plus is formed, 2 FAD H2 is formed and 2 ATP from the GTP is formed, that is called substrate level oxidation. After the substrate level oxidation, it undergoes terminal oxidation. This terminal oxidation, it is divided into 2 steps, that is electron transport chain and oxidative phosphorylation. The electron transport chain we can summarize here, that is electron transport chain, it is a chain of electron carrier that are made by iron compound, which converted into ferric to ferrous, and from the ferrous ion to ferric ion, when the electron transfer from one step to the another step. So, this is the cytochrome which is made by iron compound, as well different types of cytochromes are there. This cytochrome form complexes, as I have already discussed. 5 type of complexes are there. These 5 type of complexes, at the end of 5 type of complexes, the complex 5 involved the involvement of half molecule of oxygen and electron and proton, it converted into water. So, we can finally say that the total number of ATP is formed at the end of cellular respiration, that is 38 ATP. That 38 ATP it formed at the different places, and metabolic water. Metabolic water is formed. So, the entire reaction we can summarize here, the C6H12O6, when in absence of oxygen it form alcohol plus 2 ATP, that is called alcoholic fermentation. And next one, this is in anaerobic. Which takes place in cytoplasm. Now, the another cases, it form lactic acid, plus 2 ATP. So, in case of anaerobic respiration, the entire reaction takes place in absence of oxygen. Alcohol, that is ethyl alcohol C2H5OH is formed, and the lactic acid and 2 ATP is formed. It is found in muscle cell. when lactic acid is going to be accumulated, that cause fatigue of muscles, and the alcohol formation, it is common in yeast and in bacteria. Now, aerobic respiration. In aerobic respiration, the entire reaction C6H12O6, it combined with oxygen, it form carbon dioxide, water and 38 ATP or 36 ATP. So, when one molecule of glucose is going to be oxidized, it gives 38 ATP. So, now we are able to say that in respiration, in this chapter, there are the various types of step: glycolysis, Krebs cycle, electron transport chain and oxidative transformation. Now, I hope you will be able to answer these questions. In my next session, I will ask these questions and will give the answer. Thank you.